So I need to. Hello everyone, good afternoon all. It's Gautam and I take botany here. In the last class, we have discussed about the cell and introduction of the cell and cell theory. And even in cell theory also, we have discussed about the three important scientists there. So before going to the topic, beginning of this today's session, we will recall somewhat what we have discussed in the last session. So in the last session, what we have discussed is the previous year analysis of last 10-year question paper analysis. From this chapter, how many questions has been asked? You can see here the last 10-year analysis. There is a minimum and minimum 2 questions are there, and maximum 4 to 5 questions has been asked from this particular chapter, which marks a weightage of 20 marks, minimum 20 marks like that. So it is of that much importance chapter, even though it seems to be a basics one, basic chapter, that is cell chapter. So it has a lot of weightage and lot of importance also. See, coming to this is concept-wise analysis and where the questions can be asked the maximum in this. So we can see there from this concept they asked 48 questions from past 10 years, from here 20. The concept what we have discussed in last class, they asked around 6 questions. So next, what we have discussed in this, cell means what? What is a cell and how do we define a cell there? So that we have discussed in the last session. So cell is nothing but it is an fundamental structural and a functional unit of life. So why do we call that? I explained in the last session. If you didn't watch that one, you can go back and rewatch that video. So why do we call this cell as a structural and functional unit of life? Where you don't get that explanation nowhere, means nowhere in the sense. Uh, I explained in a detailed way so that you can go and watch that last session of botany class. So cell first session it was. There I explained why do we call that it is a structural and functional way of unit that life. Because see, we know that when we have been mucked up the statement that cell is a fundamental structural unit of life in every year, like 8th, 9th, 10th class and all. But you don't know that why do we call that and how it is connected to the functions of our bodies. So including like digestion, respiration, excretion, everything, the connections. So in the end, we need to come to the cell there. That's why I call it as this is how it is connected. So there you can go and watch in the last session where you will get a clear clarity of that. Why do we call a cell as a structural and functional unit of life? Okay. So here cells is nothing but anything less than a cell, we don't call it as a living cell or a living organism. Means to call an organism, it is a living state. So at least it has one single cell there. So taking from unicellular or multicellular organism, at least a cell should be there in an organism, so that I can call it as a living organism. Single should be there. Anything less than that, it is not a living, it is a non-living. And if you take that any one cell organism, that we call it as unicellular organism, where all the function of that, it will be taken within the same body, within the same single cell, so that we call it as a unicellular organism. And coming to this multicellular organisms, where are different types of cells we'll be seeing. See, for example, in us, to send or to pass the signals, nerve cells are there, neurons are there, which is having a long and branched structure. To transport the oxygen and carbon dioxide, RBCs are there, which is biconcave shape. So in us, there are different types of cells and functions. With respect to the functions, cell shape and sizes are different, dependent. So it is about multicellular organism. If you see in unicellular organism, all the functions will be performed within a single cell there. Okay, that based on the type of cells, we will be seeing two type of organisms, that is unicellular and multicellular organism. And coming to the scientists who gave, who worked on the cell. So first it is discovered by the Robert Hooke, that was the dead cells. And next comes an Anton von Leeuwenhoek, who discovered a living cell. And next comes Robert Brown, who discovered nucleus. And next comes another two scientists we have discussed in the last session about cell theory. So in cell theory, we will be seeing two scientists there, that is Schleiden and Schwann. These two are the scientists in 1838 and 1839 respectively, who worked on cell theory and how a cell is related to our functions and our organisms there. So here in 1838, Schleiden named a scientist who was a German botanist, and Schwann named scientist who was a German zoologist. So they gave two statements there. You already knew about this. If you didn't watch, you can just go back to the last session and you can watch there. Even though I will revise quickly here, Schleiden gave a statement that all the plants are composed of different kind of tissues and different kind of cells. So that was given by German botanist Schleiden, Matthew Schleiden. And here Schwann comes, Theodor Schwann. So Theodor Schwann in 1839, who was a German zoologist, who gave a statement that animals are covered up with an outer layer. Today it is known as plasma membrane, the thin layer around the cell. See, everything which is present here, it is a plasma. So now if I am drawing a layer there, it is the plasma membrane, because inside of this it is a plasma. So next to that, the layer is plasma membrane. So it was given by German zoologist Schwann. And next, another statement he gives, add upon to that statement, even though he is an zoologist, note my point, even though he is an zoologist, he observed plant cells and he said that plants are covered up with an extra layer that we call it as cell wall. So upon this cell membrane, there is an extra layer is also there. Today, not only today, means he only said it as it was an extra layer, that is nothing but it is a cell wall there. Okay, so it was given by him. And by seeing these two, animal cell and plant cell, and he gave an hypothesis. Hypothesis in the sense, it is an just assumption or a statement which is not yet proven by many cases. So he observed plant cell and animal cell, and he came a conclusion that this is how this it is like that. So it is an assumption made by him as a hypothesis, we call it as in scientific language. See, it is just like if you going to some place and you had breakfast, and at the same time, unfortunately, we'll get a cockroach in our breakfast. What we will discuss means what we will describe that hotel. So it is the dirtiest hotel in the whole city, and we'll comment, we'll give a negative marking that it is the worst city, means worst hotel. And based on that, by deciding that hotel, I will name the city also worst. This is assumption. I made an hypothesis that this city is worst, where the people are living in dirty places, which is unhygienic. So it is an statement, it is just a hypothesis. Means when it applies to everything, every place and every apart from not only hotels or apart from the hotels to all the places, then I can prove that it is a theory that this place is like this. So initially it will be in the form of hypothesis. So by considering this statement, Schwann and Schleiden, they together they formulated a cell theory. They formulated a cell theory, where formulated cell theory in the sense what they have given or what they added is, so all the organisms are composed of or made up of cells and products of cells. So this is formulated cell theory, which was given by Schleiden and Schwann together, that all the organisms are made up of cells and the products of cells. So add on to this statement in 1855. So add on to this statement in 1855. So another scientist comes. So his name is Rudolf Virchow or Virchow. The pronunciation doesn't matter here. So Rudolf Virchow in 1855, what he says is that this add on to that formulated cell theory in 1855, he gave modern cell theory. Modern cell theory in the sense, so we know that already that all the organisms are composed of cells and the products of cells, are made up of cells and products of cells. But no one explained, or nowhere it is explained, that how the new cell is formed there. Okay, that was given by Rudolf Virchow with the statement known as omnis cellula e cellula. With the statement omnis cellula e cellula, which means, this means, so the cells are formed from pre-existing cells. Cells are formed from pre-existing cells, means here this cell is already there. So how this cell is formed? It is formed from the pre-existing cell which was there earlier. It gave two cells. So this cell is formed from this pre-existing cell. So this statement add up on to that, it has become a modern cell theory. So this, it was given by Rudolf Virchow. So he added to that one more statement of Schleiden and Schwann. That statement was there, no? That all organisms are composed of cells and products of cells. So it was given by Schleiden and Schwann. So add upon to that statement, he gave this one. So that is omnicellula e cellula. That is meaning of that, it is the cells are formed from pre-existing cells. So the new cell are formed from pre-existing cell there. So it was given by him. Next to that, so that these questions we have already discussed. These questions already we have discussed about this one. So no need of discussing once again. We'll start the new topic today. So that is an overview of the cell. Overview of the cell means you can watch, you can have the books in front of you and see there also. See, over in the sense, complete analysis of the cell. So far what we have discussed, in the or what you have learned already in the previous classes, that about the cell. So that we are going to discuss here now. See, when I am talking about overview of the cell, I am taking a, see, this is a cell membrane. This is a cell membrane. So I am taking a cell membrane. See, within that the space is there. No, this space is enveloped of, it is composed of plasma. That some liquid will be there. That we are calling it as plasma membrane. Sorry, plasma, cytoplasma. Cyto means a cell, and the plasma means the liquid or the content which is present within the cell. It is cytoplasma. And next to that, what you are seeing, the plasma which is covering the membrane is there, no? That is plasma membrane or cell membrane, we call it as. Okay. So now this is a cell membrane which encloses this complete plasma there, or cytoplasma we call it as. Okay. So now next to this, so if it is a plant cell, So if it is a plant cell, there will be an extra membrane upon this. So there will be an extra membrane upon this. So that we call this as cell membrane, sorry, cell wall. See, initially there was one membrane was there which is covering this whole plasma there. So that is, we call this as cell membrane. Cell membrane or plasma membrane, both are same. Cell membrane and plasma membrane both are same, because cytoplasma, it is a cytoplasma whole which is present within this, it is complete cytoplasma, and the membrane which is covering plasma, we call this as plasma membrane, or it is a cell membrane, we call this as. If it is a plant cell, it will be having one more extra envelope upon this, it is a cell wall. Cell wall, if it is in plant cell or if it is in bacterial cell, so it will be a cell wall will be there, cell wall will be seen. And next to that, within this, so when you study in cell, about cell in your earlier classes, you might have come across that cell at the center there will be something will be there which is like this round structure. So that is nothing but there within that you will be seeing a, within that you will be seeing an brain of the cell. What do we call brain of the cell in the cells? So it is nucleus there. So it is nucleus, and outside of this another layer is seen. So because the nucleus is a double membrane structure, so it is, so it is a double membrane structure. So it is having pores there, nuclear pores will be there, that's why I am doing like that. So it is actually nuclear membrane, it is of double membrane nucleus it is. So it is a nucleus, and this nuclear outer layer of nucleus is there, no, it is continuated like this to form an endoplasmic reticulum. So it is endoplasmic reticulum. So it is endoplasmic reticulum. Next to that you will be seeing somewhere like this, that is Golgi complex or Golgi apparatus we call it as. So near to that you will be seeing there, so that is Golgi complex you will be seeing. And somewhere if it is a plant cell, you will be able to see the chloroplast there. So if it is a plant cell, you can see the chloroplast. If it is animal cells, or even if it is in some plant cells, it will be there, that is mitochondria will be there. So somewhere like mitochondria will be seen, mitochondria are found. And if it is plant cell, it will be having a huge structure like this which is covering 90% of the body or 90% of the cell there, which is we call it as vacuole. Central, large central vacuole will be there. So these are all organelles, cell organelles we call this as. So that is nucleus is there, endoplasmic reticulum is there, Golgi complex is there, mitochondria, chloroplast, lysosome, and even like that uh, lysosome also there, and ribosome is there. Ribosome is different, lysosome is different, don't get confused there. Ribosomes are single membrane, there is no membrane present on this ribosomes, it is just a protein and RNA complex it is. But lysosome is an, it is having a membrane around this. So in when we discuss about the cell organelles, no, so far what we have discussed, let us label that one. So that is, we'll label this all all of this will label to. So it is vacuole we know, and this is Golgi complex or Golgi apparatus or Golgi bodies, Golgi complex, dictyosomes in plants you can call. Next it is lysosome, and this one is chloroplast, and this one is mitochondria, and this one is nucleus, and within the nucleus you will be seeing genetic material there like this in the form of chromosomes where DNA will be packed like this. So DNA is packed in the form of chromosome. So it is condensed and the DNA will be present within that. And this is endoplasmic reticulum. I am writing shortly, that is endoplasmic reticulum. ER means endoplasmic reticulum. So like this many organelles will be present. So it is complete overview of the cell. And before this I said the space which is present inside of this cell, whole cell is there, no? The space which is present, all the cell organelles are present within that. That space, what do we call? That space we call this as cytoplasm. Means if nothing is there, it is just filled with some chemical there. Means that is cytoplasm. Plasma, liquid, water it is. So this, it is cytoplasm. So everything which is present within this we have discussed. It is an overview of the cell. Overview in the sense complete whatever content which is present in the cell we will be learning here. So starting from nothing is there, the cell complete, it is made up of cytoplasm, everything and all. So next to this, somewhere like in animal cells, so there you will be seeing this kind of structure. So that we call this as centrosomes or centrioles. So only in animal cells you will find this. And these ribosomes are there here. Ribosomes also is there. And this is genetic material or DNA. Genetic material in the sense which carries genes within that. So genes are nothing but from parents to offsprings how it carries. No, it is just one analogy where you can connect. But usually if you take my part of cell, so I am taking my hand cells now. If you observe under microscope that one single cell, it will be containing all these things except these two, some plant cells which carries like chloroplast, vacuole and all, except the some organelles, everything will be there. Or if you take any part of a cell of a plant, there also you can see complete organelles which we are discussing now. This is an complete overview of the cell. And in this we will be learning something special there, that is single membrane organelles. What are the organelles which containing one membrane around it, and what are the organelles which is containing two membrane around that, double membrane organelles, and what are the organelles which does not have any membrane. So that we will be seeing here in the an overview of the cell. So wait, let me make it short. So the cell organelles we have seen here. So in the cell organelles which are containing one membrane, we place that under single membrane organelles. So let me write here, that is single membrane organelles. Single membrane organelles. So single membrane organelles here are, one is ribosome. Sorry, ribosome is no membrane. There is nothing, no membrane around it. So single membrane, you can take example of vacuole. Only one membrane will be there. And what are the other organelles here? Lysosome is there. That is single membrane organelle. So these are lysosome, vacuole. So these are single membrane organelles. So there are, like that, there are double membrane organelles also. Like that, double membrane organelles also there. That double membrane organelles are like mitochondria, endoplasmic reticulum, nucleus, chloroplast. These are double membrane organelles. Like that, no membrane organelles is also there. So that is no membrane organelles. So those are ribosomes and centrioles. So that they don't have any membrane as an envelope there. So they are no membrane organelles, we call it as. So it is this data you need to remember. So what are the membrane bound organelles? What are the double membrane bound organelles? No membrane bound organelles. So this you need to remember here. So these are the things you need to remember in this overview of the cell. Okay. So now let us come to the size and shape of the cell. Whether all the cell will be having the same size and same shape, or any size differences will be there, main that we will be seeing there. So there will be some size differences will be there. So coming to that, shape and sizes of the cell. So you can see here, I will just illustrate this one here. See, in front of you, there are different types of cells are there. Different sizes and different shapes also you can see. We'll discuss one by one there, coming to the shape of the cell. Coming to the shape of the cell, so I will be writing here. Shape and size of the cell. And this shape and size of the cell, it differs with respect to the functions of the cell. For example, how can I say? See, um, even aeroplane is an transporter. It is an vehicle. And even private jet, that is also transporter vehicle. Helicopter is also transporter. Car is a transporter. Bike is a transporter. So, but the shape of each vehicle or transporter is different with respect to their function. So the aeroplane is designed in to fly in the air by applying aerodynamics principles and all. And likewise, car is designed in that way to travel, like for four wheelers, it is different. Two wheelers, it is different. So like that, if you take in cells, the cells are having different shapes based on their function. So now coming to this, one by one we'll discuss. I will show the cells later now. Okay. So coming to this, so first I will take RBC cells here. RBC cells, WBC cells, and nerve cells, or columnar epithelial cells also you can say, you can take. And smallest cells, coming to the, this is about the size, smallest cell, and largest cell, largest isolated cell, and longest cell, longest cell in humans or in animals, so that is longest cell. Or typical bacterial size also we can measure, bacterial cell. So human cell or eukaryotic cell we call it as, human or eukaryotic cell, typical eukaryotic cell size also you can take. So in general eukaryotic cell, not whether it is RBC and WBC. So coming to this, see here RBC is coming to the size of the cell. So here sizes of the RBCs will be, so it is 7 micrometer diameter. It is the diameter of RBC cell, it will be 7 micrometer. Diameter in the sense, see if it is RBC cell, the diameter of this it will be 7 micrometer. So it is the diameter. So WBC cells here with respect to the shape, here shape of RBC is biconcave shape. Biconcave in the sense both the side it is concave shape like this. So usually it will not be directly visible like this, but the biconcave shape will be like this, in which both the sides it will be having concave shape. So that is biconcave shape. When it comes to the WBC, it is amoeboidal shape. Amoeboidal cells in the sense where it won't have any proper shape there. So here you can see RBC cells. So here you can see RBC cells which are biconcave shape, biconcave in the sense, and both the sides it will be having concave. So that is the biconcave shape. If you see, it is a columnar epithelial cells. It is like columns, no? It is like columns, like this. It is columns, and it is a epithelial cells. This, regarding this, you might be learning in structural organization in animals, and their sir might be teaching there. So this columnar epithelial cells, so it is a RBC. These RBC cells which are biconcave in shape, and this one is columnar epithelial cells which is long and narrow, which is long and narrow shape it is. Now there are another type of cells are given in your book or in your module, you can check there. Mesophyll cells is given. The name with it is with mesophyll. Mesophyll cells in the sense, here you can see the diagram. So I brought you to simplify that one here. See in leaf, when you observe under microscope, you will get this kind of shape in leaf. So in this, this part what you are seeing, that is this mesophyll cell we call it as. Mesophyll cell in the sense, here in the word itself is there, phyll. Phyll means leaf, PHYLL. So PHYLL means leaf. So the leaf contains two types of cells there, usually like in dicots, that is spongy and palisade differently. In monocot it is different, spongy. So these are mesophyll cells. Mesophyll cells in the sense which are having then many chloroplasts there. So these are many chloroplasts which are present in, these are chloroplasts aligned together like so in your book what they have given, a mesophyll is given like this, a small black dots are given. So that black dots are nothing but a chloroplast. Chloroplast means which is involved in absorbing sunlight and preparing the food. You know about that. So here the mesophyll cells, the shape of the mesophyll cells shape is also differed here. So it is round and oval. The shape of the cells round and oval it is. The shape of the cells it will be round and oval. And coming to here, see this cell you can see here. This cell is a WBC cell. This is a WBC cell. So WBC cells, how it will be? See, you can see here, this structure is having like this. So with respect to this WBC and RBC and their functions, you will learn in body fluids and circulation chapter of class 11th only. So here the cell will be having a different kind of shape. So that is, it will be amoeboid shape. So some cells will be occurring like this, in some cells it will be like this. So the shape will be differed in case of WBC cells. So that's why we call this as amoeboid shape it is. WBC cells will be in amoeboid shape. And this one you know, it is very important question for 10th day. So it is nerve cell or neuron. So it is the longest cell, and arrange the shape of this one, how it looks like this. So can you think that what is the shape of that? And all the shapes it is dependent with respect to the functions of the cells there. See here, nerve cells, it is a long, so long it is, and it is having many branches like this. Many branches are there, so which is connected to the next neurons or next nerve cells there. So it is having branches. So nerve cell shape is long and branched. So you need to remember all the shapes here because it is very important for NEET aspect. See here it is long and branched. It is long and narrow. Don't confuse it in these two. Okay, this shape is long and narrow here. So this is columnar epithelial cells, and this one is RBC cells, and this one is mesophyll cell. So that is mesophyll cell, and this one is WBC cell, and this one is nerve cell. And there is one more cell is given, only this shape they have given in your book, but I wanted to give that clear picture what cell it is and where it is present. So that's why I gave this plant analogy here. See, if you cut the plant, it looks like this, cylindrical. So it is a plant. When you cut that one, it looks like a cylindrical. So within that, it will be having some xylem and phloem will be there because the water has to move, no? So the water has to move upward like this. So where the water transportation will be taking place. So that it will be done with the help of xylem. So xylem will be having elements there. So xylem is made up of 4 elements, that is tracheids, vessels, and xylem parenchyma and xylem sclerenchyma or xylem fibers. So here it is having tracheids. What we are going to discuss now, the shape of that is tracheids. What we are going to discuss is this one, that is tracheids. Like this, these are there. No? So these are tracheids there. So this, so in this, what we are going to discuss is tracheids. So tracheids shape is somewhat elongated, and it will be tapering end. See, you can see this one. If you see this one, it is having blunt end it is. If you see in this one, it is having narrow like this and it is ending like this. This is, we call this as a tapering end in the sense where they will be colliding and making conical like structure there. So tracheids, it is having the shape of elongated shape. Now coming to this part, why we are learning all these things and the shape. So we need to analyze the functions also. RBC cells are biconcave in shape. Why? Because there will be no nucleus in the cell, and they are helpful for transportation of oxygen and carbon dioxide. See, you just inhale. When I am inhaling, I am intaking the air, and that it reaches to the lungs, and the oxygen it will be there when it reaches to that part. So there will be exchange. These RBCs are the vehicles where the oxygen they will sit back of that, and they will take that to the different parts of our body wherever it is required. So RBCs are developed, or RBCs are having this shape based on their function. Their function is to transport the oxygen. So there, based on the function, it is of so it is based on the function, the shape of the RBC is different. And coming to this columnar epithelial cells, so it is a long and narrow. So it is long and narrow here. It is long and narrow. And coming to the mesophyll cells, it is round and oval. It is round and oval. Mesophyll cells are round and oval. And coming to this WBC cells, they are amoeboid in shape. Coming to this nerve cell, see, we know that nerve cells are, so their nerve cells functions, we know that the function of nerve cell. Function of nerve cell is to send and receive the signals. So if I am pinching here like this, the nerve cells will be sending the signal. So to send the signals, how a cell should be? So the shell should be in the form of long and branched. So if it is branched, it can pass the information very quickly. So like that, the cell is modified their shape based on their function. So their function is to send the signals and receive the signals. So that's why it is long and branched here. Coming to the tracheids. So you know that xylem, the functions of trachid is to supply the water. So to transport the water. So when it is transporting the water, it has to be required in the form of pipe. So the pipe, how it will be, in the same shape, it will be elongated, it will be having like this. So the elongated shape, the tracheids will be. So it is about the shape of the cell. And coming to the size here, the smallest cell we are talking about here. So nerve cell is the longest cell. It is the longest cell. And the smallest cell is mycoplasma there. Mycoplasma, we call this as smallest cell. So smallest cell in the sense mycoplasma, so which measures around 0.3 micrometer length. It is a length. So even it is important whether it is length or diameter and all. Here it is a diameter 7 micrometer. Mycoplasma is of 0.3 micrometer length it is. So largest cell, largest isolated cell is ostrich cell. Ostrich egg cell. So longest cell is nerve cell here, which I already wrote there. So longest cell is nerve cell, and bacterial cells you need to see here. So bacterial cell, which is in your book they have given different there. So but coming to the length, it will be around 3 to 5 micrometer length. The length of bacteria, it is 3 to 5 micrometer, and Coming to the human eukaryotic cell or typical eukaryotic cell, it will be measured at 10 to 20 micrometer. So 20 to 20 micrometer it will be the human eukaryotic cell. Or even in some books they have given 10 to 100, but you just refer the NCRT data, that is 10 to 20 micrometer it will be there in case of that. Okay, coming to the virus. So virus, it is different again. So virus is not a living cell, we call this as, but when it is coming in contact with the host, so it will be living there. So virus, which is measuring around 0.02 to 0.2 micrometer, so that is the size of virus. So you might be getting question there. I am saying the smallest cell is mycoplasma, which is of 0.3 micrometer. So you might be getting question that why virus is not the smallest one even though it has 0.02 micrometer. So the answer is very simple. I said in the last class also, and even in the beginning of the class also I said anything less than a cell is not a living organism. So if you see this virus, if you see this virus, if it is present on this pen, it is a non-living. Virus is a non-living because the virus does not have a proper cell there, cellular structure. So the virus is non-living. And when that virus get to know that I am scolding that you are a non-living cell, non-living fellow, then when I touch and inhale, so now it become alive. So it become a living cell now. The virus will turn into a living when it come in contact with the host cell, either it is a plant or animal. So that's why we cannot consider virus as the smallest living cell. So smallest living cell is mycoplasma there. Even in the book they have given that another data is given where PPLO is added. PPLO is nothing but it is a type of mycoplasma which has the size of 0.1 micrometer they have given. So it is the average typical size of that PPLO. It is a type of mycoplasma there. Don't get confused there. It is the length, it is 0.3 micrometer length. But if you look at the size, it will be of 0.1 micrometer. Usually it will be. So here what we have discussed is the size and shape of the cells now. So in today's class, whatever what we have discussed so far is, so this one cell, overview of the cell we have seen, and single membrane, double membrane and no membrane bound organelles we have seen. So a revision, quick revision of cell theory and modern cell theory we have seen, and on function based how the cell shape and sizes are differing we have seen. So we'll solve some questions related to that now. That is, here it is. Which of the following cells have diameter of 7 micrometer? So if you are watching live, you can comment down there. Which of the following cells have diameter of 7 micrometer? So in your book and in your modules only diameter word is used for only one, that is nothing but here what is the options are given. Just read all the options first and go for the marking. Erythrocytes, monocytes, neuron and blood platelets. So here erythrocytes is given. So erythrocytes is nothing but erythro means what? Erythro is this word refers to red there, red blood cells. Cyte in the sense cyto or cytes in the sense cells. So it is a red blood cells. So these are RBCs there. So if RBCs are these, so RBC is having this one. So the correct answer is this one. Okay. Arrange the following cell in ascending order of the cell. Based on the size, you need to ascend that, so that in ascending manner, mycoplasma, ostrich egg, human RBC, and bacteria is given. So you know the length of this one and the sizes of the different cells there. So you can place that in ascending order. So we know that ostrich egg is the largest cell. So then T should second should be, the second option should be at the end. Okay, so that is here. Next we'll go for smallest cell, that is mycoplasma. So it is at the first. So remaining 4 and 3 are there. So 4 is bacteria and 3 is human RBC. So RBC is 7 diameter, 7 micrometer. Bacteria is 3 to 5 it will be. So which one will be the answer? So this one will be answer. 1, 2, that is 1, 4, 3, 2 will be the answer. So I hope you enjoyed the session. Is there any other question? Okay. So next, in the next class, we will be learning about the prokaryotic cell and eukaryotic cell and the types of cells based on the organization of the cell. So I hope you enjoyed this session, and we'll see you in the next class with different analogies and with examples. We'll learn with fun, and where you can remember and understand in a conceptual way, where you can link to the your own life. Even though we will be discussing prokaryotic cell, don't mind that it is difficult to imagine and all. So I will make you to connect to the real life, and till that, keep tuning. Thank you.